And our next talk is by Dr. Stephen Abaddon from The Ohio State University, titled Ecology of Bacteriophages as Anti-Biofilm Agents.
Hello. Okay, so this might be a little weirder than usually when I give my talks because this is me last night in the emergency room having wiped out my motorcycle. I'm still trying to figure out whether the motorcycle is functional. But anyway, I've got 12 stitches. So I have to describe what I am because otherwise this talk will be even weirder than it already is. So this is me. So I'm sort of a theoretician. I make a lot of conceptual models. So I think about stuff. I combine things. I synthesize. And I also use computers to do some math, although I'm not a mathematician. But my training was in the lab. So when I'm doing mathematical stuff, I tend to want to do something that's at least an experiment as I perceive it on a computer. I don't have students. I don't have time to do my own experiments. But what I do is I sort of pretend to be an ecologist. And I've become sort of pretending to be a pharmacologist. And I've been doing history papers for a long time. And also I do a lot of teaching. So I study bacteriophages. I'm a phage ecologist of sorts. Bacteriophages come in a number of different shapes and sizes. I tend to emphasize looking at tailed ones just because that's my habit. But there are other types of phages out there, including filamentous ones. They're not as diverse morphologically as the archaeoviruses. I'll talk about archaeoviruses. But they're particularly weird, in part because we're only now just studying them. I'm interested in the interaction, one of my interests is in the interaction between phages and biofilms. And so this is just a model I put together that's standard of biofilms. So biofilms involve bacterial attachment surfaces. They involve some kind of growth. And they involve some kind of dispersal. It doesn't necessarily look like that. And that'll be important as we get to the end of this talk. The phages care, I think, mostly about that part. The bacteria care mostly about that part. The whole idea is to find a place to make more bacteria so that ultimately they can make more bacteria or find a place to make more bacteria. I'm also interested in phage therapy. That's where the pharmacology comes in. Phage therapy is the use of bacterial viruses to treat bacterial infections. It's been with us for approximately 100 years now. When do we use, or when should we be using phage therapy? You can use it anytime you want, really, if it's available. But mostly it's thought of as something you use when you have antibiotic resistance infections, you need something that'll work. Or antibiotic tolerant infections, where the bacteria aren't actually genetically resistant, nonetheless the infection is not responding to antibiotics. Or indeed, if you want to spare your microbiome, that would be a good idea, a good reason to use phages, since they have narrow host ranges, narrow spectrum of activity. I went to Google Scholar and tried to get a handle on the use of the phrase phage therapy and quotes. In the late 1990s, roughly 20 years ago, we started to have this exponential increase. And then there was a pause, apparently, and now we're on another exponential increase, and we're doubling in terms of its usage in literature, according to Google Scholar, at a rate of about 4 years. Why use phage therapy? Well, phages are bactericidal, not all antibiotics are. Phages can replicate when they're in contact with bacteria in situ, so increase their dose in situ. They have a low inherent toxicity. They have a low impact on normal flora. Narrow resistance evolution means if you're not infecting all the bacteria out there, then there are only a limited number of bacteria in which resistance can actually evolve. Lack of cross-resistance with antibiotics. Easy to discover. They can be used with other agents such as antibiotics. There's a versatility of dosing. You can clear biofilms with them, which I'll get back to. And they have interesting pharmacokinetics that seem to work better than you would think they would. And then there are other reasons, but I'll skip over those. I put out a paper last year where I was trying to understand the phage impact on biofilms versus the impact of antibiotics from the perspective of their ecology, what's going on in the real world. I made the argument that phages are potentially superior to antibiotics as antibiofilm agents because Some of them can actually hydrolyze extracellular polysaccharide. They have single hit kinetics. They actually concentrate their activity. This is that auto-dosing in association with bacteria they're attacking. They lyse bacteria, they're bactericidal. They increase their application in situ, their concentration in situ, and they evolve to be obligate predators of bacteria, where these things tend to be lacking in antibiotics. So as a consequence, though antibiotics certainly can have biofilm activity, they're not necessarily have to have the same potential as phages to clear biofilms in a clinical sense. Oops, wrong one. So this is trying to state that phages have their problems also. In thinking about phages interactions in nature, but also phage interactions in the clinic, what is limiting the ability of phages to attack, especially biofilms? So phages might be limited in their ability to move into biofilms and reach bacteria. They might be limited in their ability to move around the biofilms, go from bacteria to bacteria within the biofilms. They might be limited in their ability to move out of biofilms. Ultimately, it's the number of phages out of biofilms that's going to determine the potential for phages to control biofilms in nature and also in the clinic. And these numbers are probably limited for reasons that I'll go into. So this is another way of looking at phage biofilm interactions. This is a spatial scale, so we're going from small target bacteria, clones of bacteria, whole biofilms to the larger environment. And this is showing the same idea, only much more complicated. So this is how this idea maps onto these. So that's a single cell, that's a microcolony, that's the biofilm. And the larger environment is that. So that's another biofilm, basically. And my model, which I published a couple of years ago, the idea is that phages may be limited in their impact on bacteria in the wild because they in fact have, biofilm bacteria in the wild, because they have trouble, in fact, amplifying their numbers to sufficient densities to, in fact, even keep up with the elimination of these phages by inactivation, especially between biofilms. So a single phage finds a bacterium and infects, releases phages. Maybe those phages are able to find other bacteria that are close by within the same microcolony. Maybe they're able to diffuse within the biofilm and find bacteria that they're able to infect elsewhere in the biofilm. Collectively, they're able to build up a certain number of phages that are able to, in part, leave the biofilm. And then these phages are the ones that are charged with finding new bacteria, new microcolonies, new biofilms. These phages infecting here have to build up their numbers to a sufficient amount that the numbers that actually go exploring outside of the biofilm can survive to the point where at least one of them is able to find new bacteria and biofilm microcolonies to infect. So from the phage perspective, they generate enough barriers to survive their extracellular search, I should say extra biofilm search, to find new microcolonies to infect. From the bacterium, from the bacterium's perspective, larger, more numerous, more readily reached, less phage-resistant microcolonies being more impacted by phages. So this is thinking about phage biofilm interactions as an ecological steady state. These circles here are kind of like plaques. So a phage reaches a biofilm and starts replicating. Maybe the phage finds more bacteria to infect. At a steady state, the number of phages that are produced per these foci of infection is basically enough so that all but one of them dies before finding a new bacterium to infect. That's steady state. Now, if the population is growing, of course, there'll be more that survive. The population of phages is declining, few of them will survive. But at steady state, they're doing everything they possibly can just to get enough phages released from the biofilm to actually just hold on, just have one phage start the process successfully all over. So phages collectively probably are not having a huge impact on biofilm bacteria in nature because they're probably just surviving, except perhaps for single species biofilms. But even then, there's probably limitations to the ability of phages to actually propagate within biofilms within microcolonies. I'm starting to explore this idea of a heterogeneity in the physiology and structure of microcolonies and biofilms, especially microcolonies, in terms of their ability to prevent or support phage replication. And so you have a phage that is absorbing, and then this phage is preferentially going to be absorbing to bacteria on the outside of microcolonies just because they're more available spatially. These bacteria on the outside probably are also better able to support phage infections. And on the outside, the EPS, the extracellular polymeric substance, is probably less mature, thereby potentially allowing the phages better access. In the interior of the microcolonies, the EPS will be more mature, the bacteria less capable of supporting phage infections. We can then move on to thinking about bacteria in a steady state. So this is a bacteria-initiated microcolony. The microcolony needs to make enough bacteria to release those bacteria so that at steady state, at least one of those bacteria finds its way into a spot where it initiates its own successful microcolony. You can also think in terms of the bacteria fighting back against the phages. There are a number of mechanisms, not all of which are very robust, that are able to prevent phages to some degree from propagating through microcolonies. Ultimately, the goal of phage resistance within the context of the microcolonies is presumably towards eventual biofilm bacterium dispersal. In terms of the phages, we can think of them as herbivores. The phages, in fact, are infecting these microcolonies and perhaps not eating all of the microcolonies, just the tender young leaves rather than the twigs. Moving on to phage therapy. So this is a treatment of staphylococcus-infected diabetic ulcers. It's not my work. Basically, these patients were told, we're either going to cut your toe off or you're going to get some kind of treatment. They did this treatment. This is treatment with phages. And you see improvement in this next example, even better improvement. These treatments lasted a fairly long period of time, 8 weeks, and they took multiple doses. It is not easy to get rid of biofilms with phages, but it may indeed be possible to get rid of biofilms in the clinic with phages. I record all my talks. Hopefully this one's being recorded right now. You can find them in numerous locations. The easiest one is probably phage.org. Just click on the author button. I came back from England on Tuesday morning at 2 a.m. and was totally out of it, so spent all day Tuesday updating my website. I'm waiting for that finger that's up in the air to go down. And that's the end of my talk. Thank you very much. Yes? Another advantage of phages is that they tend to be self-limiting, so they don't stick around. People are trying to create materials that have phages impregnated in them so they can stick around. But yeah, phages tend to go away. The other thing that you didn't mention is that there's some potential for an immune response. But that doesn't seem to be as big a deal as people like to think it is. There's a paper that recently came out that basically argued that immune response against the phages doesn't seem to have an impact on phage therapy. No. Okay, thank you very, very much.